AS Level Biology 9700 Paper 2 Variant 1 October November 2025 Question number 1 and 2 and we are moving towards the question number 1. Question number 1 is Figure 1.1 shows a diagram of the fluid mosaic model of the structure of the cell surface membrane. You can see the phospholipid bilayer cell surface membrane in which this is the phospholipid molecule, head and tail. Head is phosphate, which is hydrophilic, water-loving, and linked with glycerol. And tail is hydrophobic, water-hating, so fatty acids. So we have to label the parts A, B, and C. First of all, you can see the A. A is a carbohydrate. You can see the 6 carbon over there. So this is the carbohydrate molecule. So carbohydrate, in this case, the carbohydrate molecule is attached on the round head of the phospholipid. So it is glycolipid. It is glycolipid. A is glycolipid. And second one is a small hydrophobic molecule tucked between the phospholipid tails regulating membrane fluidity. You can see over here, it is between these two phospholipid molecules. So this molecule is the cholesterol. So this is a small molecule, it is cholesterol. And the C, again, this is the carbohydrate chain attached with a large globular shape. And this globular shape is protein. So this is glycoprotein. C is glycoprotein. Next part of this question is draw an arrow on figure 1.1 to show the movement of a molecule across the membrane into the cell by facilitated diffusion. So, you know that the cell surface membrane acts as a boundary which will not allow all the molecules to pass through it. Only larger molecules will be forbidden to pass through. So they need proteins, transporter proteins, carrier proteins to move through. So here in this case, we can draw arrow in this direction. And C part of this question is explain how the fluidity of the membrane would change if the proportion of unsaturated fatty acids in the phospholipid bilayer increased. Students, you know that if fluidity increases, the proportion of the unsaturated fatty acids will make the membrane more fluid. So how can they become more fluid? Because you know the unsaturated fatty acids contain carbon to carbon double bonds. So these double bonds create kinks or bends in the hydrocarbon tails. So these kinks prevent the phospholipid molecule from packing closely together. So this increases the distance between the molecules, reducing the intermolecular forces. So if we go with it, then I think I should write this answer as well. increasing the proportion of unsaturated fatty acids will make the membrane more fluid. More fluid, so because they contain carbon to carbon double bond. So these double bonds creates bends or kinks in the hydrocarbon tails. So these kinks prevent the phospholipid molecule molecules from packing closely. We're packing closely together. This increases the distance between the molecules. Between the molecules, reducing the intermolecular forces. Between the tails. Now, the next question is, and you know the saturated fatty acids have single bond. So here question number 1 part D is, the R groups of amino acids give them different properties. So suggest position 1 of P may differ from the R groups of the amino acids at position 2. Moving towards the figure, you can see this is the position 1 and this is the position 2. So you can see the position 1, the R groups are likely hydrophilic. So this allows them to interact with water or polar molecules. Whereas the position 2, the R groups are likely hydrophobic. So non-polar, so they allow them to stay stable within the lipid environment or the core of the protein away from water. Because the position one is outer side and position two is inner. So we can say that if this diagram shows a different arrangement, so I would go the if you like the answer at this given space, which is of only 1 mark. Position one, the R groups are likely hydrophilic. They this allow them to interact with water. And position two, the hydro groups, the R groups are likely hydrophobic. Hydrophobic. So this allow them to interact. Uh, this allow them to stay stable within the lipid environment. Away from the water. Moving towards the next question. And next question number 2 is hemoglobin is a globular protein containing heme groups. Explain how the presence of heme group allow the hemoglobin molecule to transport oxygen. And students, you know that the hemoglobin has 4 chains because it is a globular protein which consists of 4 polypeptide chains, 2 alpha chains and 2 beta chains. Each chain contains one heme group, and heme group make a bond with the oxygen molecule. So I would like to say that the heme group is a prosthetic group. So it contains uh, it allows for oxygen transport. So each heme group contains an iron at its center. So this iron has affinity for oxygen and can bind to one oxygen molecule. So one hemoglobin molecule consists of 4 polypeptide chains. So each with its own heme group, a single hemoglobin molecule can carry up to 4 oxygen molecules. So the bond between the iron and the heme group on the oxygen is weak enough that it can easily be broken. So this allows oxygen molecule to bind to high partial pressure environment of the lungs and be uploaded in the low partial pressure environment of the respiring tissues. So in this way, we can say one heme group binds to oxygen, the entire hemoglobin molecule changes shape, making it easier for the remaining heme group to bind to oxygen. So oxygen is not very soluble in blood plasma. By binding it to the hemoglobin, the blood's oxygen carrying capacity is increased by roughly 70 times. So after too much explanation, I would like to now, because the answer question is of too much, so we have to write it shortly on the given space. So hemoglobin consists of four chains having heme groups. Each heme group contains iron ion at its center, which has for oxygen and can bind to one oxygen molecule reversible. The bond between the iron and oxygen is weak enough that it can easily be broken. This allows oxygen to bind with hemoglobin with it. So when it when one heme group binds with oxygen, the entire hemoglobin molecule changes shape. Entire hemoglobin molecule changes shape. making it easier for other heme groups to make bond with oxygen. So in this way, it is the hemoglobin molecule transport oxygen. Now the part B of this question is, in certain situations, such as during intense exercise, a person may breathe very quickly and deeply. This is known as hyperventilation. Since this hyperventilation causes more carbon dioxide to be exhaled. As a result, there is a lower concentration of carbon dioxide in the blood that passes through the capillary network in respiring tissues. This increases the affinity of hemoglobin for oxygen so that there is a decrease in the release of oxygen from red blood cells. Explain why a decrease in the concentration of carbon dioxide in the blood passes through respiring tissues leads to a decrease in the release of oxygen from red blood cells. So I will go with this question, that is uh, the formation of, the carbon dioxide typically reacts with water inside red blood cells to form carbonic acid catalyzed by the enzyme carbonic anhydrase. So this acid dissociates into hydrogen ion. So carbon dioxide, when reacts with water, so it will dissociate carbonic uh, into carbon dioxide, carbonic acid catalyzed by the reaction carbonic anhydrase. So this acid hydrogen ions and hydrogen carbonate ions. So when carbon dioxide levels are low, this reaction shifts to the left. Consequently, there are fewer hydrogen ions produced in the red blood cell. So hydrogen ions normally compete with oxygen for binding sites for hemoglobin. When hydrogen binds to hemoglobin, it forms hemoglobionic acid, so which reduces hemoglobin affinity for oxygen and causes it to release oxygen. So during hyperventilation, since there are fewer, less hemoglobin I formed, so hemoglobin maintains a high affinity for oxygen. So the hemoglobin is holding onto the oxygen more tightly, so it does not dissociate as easily. Therefore, even the respiring tissues need oxygen. The hemoglobin does not release into the plasma to diffuse into the cells. So we have to write the answer. Lower carbon dioxide concentration means less carbonic acid is formed and pure hydrogen ions are produced. Are produced pure hydrogen ions means less hemoglobinic acid. is formed which lowers the pH. So the pH inside the red blood cell remains high. This results this results in hemoglobin having a higher affinity for oxygen. Means meaning the oxyhemoglobin dissociation dissociation curve does not shift to right, and oxygen is not released. At the tissues. So, moving towards the next question, part C. And you can see the part C is a diagram. Figure 2.1 is a diagram of a section through the heart. Here you can see atrioventricular node and porcine fibers. So the X describe the and explain how the tunica media, middle layer of the wall of the blood vessel X, adapts the blood vessel for its function. So this is the blood vessel, adapts the blood vessel for its function. So X is aorta. So X is aorta. So as the main artery leaving the left ventricle, it must handle extremely high blood pressure. So the tunica media is specifically adapted to manage these physical demands. So what are the structural adaptations? So we have to write the structural adaptations of tunica media. So one of them is that is the tunica media in the of the aorta is notably thick and contains a high proportion of elastic fibers. So and smooth muscles. So elastic fibers allow the wall to stretch significantly when they left ventricle contacts and pushes a surge of high pressure blood into the vessel. So smooth muscles, they have strength and control, so they provide structural strength to the vessel. So it prevents the artery from bursting under high pressure, while the aorta is primarily elastic rather than muscular. Compared to small arteries, the smooth muscles still allow the same vasoconstriction and vasodilation to help regulate blood distribution and pressure. And third adaptation, they have collagen fibers, which provide tensile strength, acting as a mechanical limit to prevent the vessel from overstretching and tearing. So we have, first of all, uh, we will go with, that is the axis aorta. So tunica media of the aorta. Aorta is notably thick and contain a high proportion of high portion of elastic fibers and smooth muscles. The elastic fibers allow the wall to stretch significantly significantly when the left ventricle contracts. So left ventricle pushes the blood towards the aorta, and aorta distributes the oxygenated blood throughout the body. So it needs uh, more elastic fibers, and you can see the smooth muscles. And the second type of the muscles, I have told you they are the smooth muscles. Smooth muscles provide structural strength to the vessel wall. So it prevents the artery from bursting. And collagen fibers provide tensile tensile strength. Now the last part of this question is, describe the role of the atrioventricular node and the porcine tissues in the cardiac cycle. So Atrioventricular node acts as a gatekeeper between the atria and the ventricle. Its primary role is it picks up the waves of excitation that spreads across the atrial wall from the synatrial nodes. So AVN shows down the electrical impulses for a fraction of a second. So uh, its purpose of delay is crucial because it ensures the atria having finished contracting and having fully emptied their blood into the ventricles before the ventricles high to contact. Then porcine tissues, once the impulse leaves the atrioventricular, it travels down the septum via the bundle of hairs and into the porcine tissues. So porcine tissues consist of specialized muscle fibers that conducts the electrical impulses very quickly to the apex of the heart. So the fibers spread out through the walls of both ventricles. So I'm going to write well. Role of atrioventricle. First of all, the role of the atrioventricular node. So atrioventricular node picks up the wave that spread across the the so the atrioventricular node slows down the electrical pulse for a fraction of second for a fraction of second. So this delay is crucial because it ensures the atria have finished contracting. And fully emptied their blood into the ventricles before the ventricles begin to contract. And then the role of the porcine tissues. The porcine tissues consist of smooth muscles. So the fibers spread out porcine spread out through the walls of both ventricles. By delivering the impulse to the apex first. The protein tissues ensure that the ventricles ensures that the ventricles contract from the bottom upwards. This pushes blood efficiency efficiently out of the heart into main arteries. Main arteries. So it was all about question number 1 and 2. Please like, share, and subscribe my channel. Thanks for watching.